All right, welcome. Today we are diving deep, really deep into our biological instruction manual. We're going to solve some really cool genetic puzzles, and believe it or not, we're starting in the grocery store and ending with the code that literally makes you, well, you. So let's kick things off with a question you've probably never even thought to ask. Why are the bananas you get at the store, perfectly seedless? It sounds like a small thing, right? Just a convenience. But it's actually a real genetic mystery. And cracking this one open, it unlocks some of the biggest, most fundamental ideas in all of biology. Seriously. Okay, so before we can solve the great banana mystery, we've got to get on the same page about the rules of the game. We have to understand how life passes its instruction book, its DNA, down to the next generation. And it all boils down to the two different ways our cells can split. All right, you can think of it like this. You've got mitosis, which is basically a biological Xerox machine. It just makes perfect, identical copies of cells for things like growth and repair. Simple enough. But then there's meiosis. Meiosis is the interesting one. It's what makes reproductive cells, sperm and eggs. And it's less like a copy machine and more like shuffling a deck of cards. It's all about creating unique genetic hands. And for our story today, meiosis is the process we really need to wrap our heads around. Okay, so let's really dive into meiosis, because it's not just a simple 50-50 split. No way. It's this beautifully precise cellular accounting system. It has super strict rules that honestly make all of sexual reproduction even possible. So here's the playbook for meiosis. You start with what's called a diploid cell. That just means it has two full sets of chromosomes. One from mom, one from dad. The first big move, meiosis I, separates those matching pairs. Then, in the second step, meiosis II, it splits what's left. But here's the most important part, the whole goal. You end up turning that one starting cell with 46 chromosomes into 4 totally unique haploid cells, each with exactly 23. Perfect. And this is the absolute key takeaway. Meiosis is a game of precision. The entire point is to make sperm or egg cells with exactly half the genetic info, not one chromosome more, not one less. Any mistake in this division, which has a fancy name, non-disjunction, can lead to some really serious issues. The system has to be, for all intents and purposes, perfect. Okay, so now that we know the rules, let's get back to our first puzzle. The rule is simple. Perfectly halve your chromosomes. So, how in the world does the banana you had for breakfast just completely throw that rule out the window?
And here it is, the big reveal. The common commercial banana, it isn't diploid. It doesn't have 2 sets of chromosomes. It's triploid. It has 3 complete sets. We often write that as 3N.
Right, so this condition has a name. It's called polyploidy. And it's actually super common in the plant world. It's why we have things like giant strawberries. Some of them have 8 sets of chromosomes, or even sugarcane. But in animals, not so much. It's extremely rare. So, why does having 3 sets of chromosomes make a banana seedless? Well, it completely breaks meiosis. Remember how we said chromosomes have to pair up to divide correctly? Imagine trying to pair up dancers when you have 3 of everything. There's always going to be a third wheel left out. This makes the whole separation process a complete mess, just chaotic. The gametes that get made end up with a totally random, jumbled number of chromosomes, which means they're just not viable. And if you don't have viable reproductive cells, you don't get viable seeds. And there you have it. That's why your banana is seedless. Mystery solved. All right, pretty cool, right? So we've cracked a puzzle about the number of chromosomes. Now let's shift gears and look at a different kind of mystery, one that's about specific types of chromosomes. And this brings us to the calico cat. You know, the ones with those beautiful, distinct patches of orange and black fur. But have you ever stopped to think, why are they almost always female? It's not a coincidence. The answer is hiding in our sex chromosomes, the X and the Y. The secret here is an amazing biological process called X inactivation. So here's the deal. In mammals, like us, females have 2 X chromosomes, and males have an X and a Y. The thing is, the X chromosome is packed with critical genes. Having 2 active copies running at the same time would actually be too much of a good thing. It'd be toxic. So nature has this brilliant solution.
In every cell of a female, one of the X chromosomes gets randomly shut down or inactivated very early in development. It's still there, but it's just silenced. And believe it or not, that is how you get a calico cat. It turns out the gene for orange or black fur in cats is located on the X chromosome. So if a female cat inherits one X chromosome with the orange gene and one with the black gene, X inactivation goes to work. In some patches of cells, the orange X gets turned off, so those cells make black fur, and in other patches, the black X gets turned off, so they make orange fur. The result is this beautiful living mosaic of color. And because male cats only have one X chromosome, they can be orange or they can be black, but they can't be both at the same time. Okay, so we've solved a plant puzzle, we've solved an animal puzzle. Now let's bring it all home. How does this kind of thinking apply to us? How can we use this deep knowledge of genetics to understand and even predict our own genetic future? Well, one of the most powerful tools geneticists have for this is something called the Hardy-Weinberg principle. You can think of it as a kind of mathematical ruler. It gives us a baseline to predict how common certain genes are in a big population, as long as that population isn't evolving. It's a real cornerstone of population genetics. Let's make this real. Let's use it to look at Tay-Sachs, which is a recessive genetic disease. So we start with the variable Q. That's just the fancy term for how often the recessive allele for Tay-Sachs shows up in a given population. Let's say it's 0.017 or 1.7%. Easy enough. That means P, the frequency of the dominant healthy allele, has to be everyone else, so 1 minus that, which is 98.3%. Now, what if we want to know the chances of being a carrier, that is, having one copy of each? For that, we use the simple formula 2 times P times Q. And when you plug in the numbers and do the math, you find out that the probability of someone in this population being a carrier is about 0.033, or, to put it another way, about 1 in every 30 people. And you can see how this would be just incredibly powerful information for things like genetic counseling. But wait, Hardy-Weinberg is for big, random populations. What if we want to get personal? What if we want to calculate the risk for specific people in a specific family? For that, we need a different tool, a family tree, or what geneticists call a pedigree. So let's pose a new question. In a family that we know has a rare recessive trait floating around, what's the actual probability that two cousins have an affected child? The way we solve this is by building what's called a chain of probability. You have to trace the gene through the family tree, step by step. We won't go through the whole diagram here, but let's just say we've done the work and we've figured out that the probability of the first parent being a carrier is 1 in 3, and the probability for the second parent is also 1 in 3. Now, we also know from basic genetics that if both parents are carriers, there's a 1 in 4 chance their child will actually have the condition. To get the final, overall probability, you just multiply all those chances together. So what do we get? 1/3 times 1/3 times 1/4 gives us a final answer of 1 in 36. Just like that. By carefully applying these rules, we went from a big population-wide guess to a super specific risk for one future child in one family. That right there is the predictive power of genetics. I mean, just take a step back and think about the journey we just took. We started in the produce aisle, asking about a seedless banana, and we followed that single thread all the way down to the deepest levels of our own genetic code, calculating the odds for our own families. And that brings us to a final and maybe the most important question of all. We have gotten incredibly good at reading the code of life. We can look into our genetic past and predict our genetic future with more and more accuracy. So the question really isn't, can we know anymore? The real question is, what should we do with that knowledge? And that is something to think about. Thanks for joining me.